Okay, next speaker will be Jamil Akim, and I asked him to add my name here. And Jamil is the best intern I ever had, and he spent this hot summer in dark place, generate about 2 giga of image, and he wants to show a part of his image today. And his topic is a biopsy analysis of a temporal interaction of a lipid biosynthetic enzyme. That is from the Castelline. Jamil.
Thank you, Shinjin, for the introduction. Thank you, ladies and gentlemen, for coming out today. So, as Shinjin said, the title of my project is Biopsy Analysis of Temporal Interactions of Lipid Biosynthetic Enzymes. So my objective of my research this summer is to confirm results from the yeast-2 hybrid assay by performing time course analysis of protein-protein interactions using bimolecular fluorescence complementation in living tobacco cells. So here is an interaction map of biosynthetic enzymes that my mentor Shinjin previously tested. These biosynthetic enzymes are known to be involved in lipid biosynthesis in plants. And these enzymes were tested using the yeast-2 hybrid method and also biopsy analysis. We identified that positive reactions found in the yeast-2 hybrid could not be confirmed using biopsy analysis. So Shinjin performed the experiment using biopsy analysis after the 3rd day and couldn't find any interactions using biopsy analysis. So we hypothesized that some protein-protein interactions may be time-dependent. And also, so we designed a time course experiment to observe protein complex expressions in tobacco cells using a confocal microscope. So what is biopsy? Bimolecular fluorescence complementation is a method typically used to validate protein-protein interactions. So protein-protein interactions occur when 2 or more proteins combine together to carry out a biological function. So here we have a YFP fragment, YFP yellow fluorescent protein fragment, which is split into 2, which is linked to 2 proteins of interest, proteins A and B. And if a interaction between the proteins when the proteins are fused together and an interaction occurs, then we have a bimolecular fluorescent complex, which shows using the confocal microscope. So a few methods for detecting protein-protein interactions are, as we use the bimolecular fluorescence complementation, the yeast-2 hybrid method. Also, we use fluorescence resonance energy transfer, bioluminescence resonance energy transfer, and also co-immune precipitation. So why do we choose biopsy? Biopsy is a great method to use in visualizing protein-protein interactions and subcellular localization of protein-protein interactions. It validates protein interactions, and you can validate protein interactions by using the confocal microscope. So here are some examples of a positive and a negative reaction. Here we can see our YFP fragment, which is fused to the proteins of interest there in the positive reaction. Here. And D is red, our expression vector, which tells us that if the cell has been transformed or not. And here is a merged image of the YFP and the DS red together. And here you can see a yellow fluorescent complex, which indicates to us that an interaction has taken place. Also here is a negative interaction of 2 proteins. And we indicate here that you might see background fluorescence here. And that just indicates that a negative reaction has taken place. We can't see the nucleus. Right here is the nucleus, and we can't see that here. It's very slight, but in the YFP fragment, is very, very minimal. So this is how we can determine if a negative reaction has taken place or not. Here is another interaction map which shows our proteins of interest that we tested, and we found that 2 proteins, DGAT2 and PDAT2, interact with both OLE. And DGAT is diacylglycerol acetyltransferase, and the OLE is oleucin. Sorry, and the PDAT is phospholipid diacylglycerol acetyltransferase. So among all the proteins that we tested here, proteins we found an interaction, and this is a chart showing the intensity of the interaction here. So FH12, CYTB5 is our control protein that we use. These two proteins are known to have a positive reaction when they are fused together. So this is why we use it as our control. And as you can see, the first day, second day, and third day, and even the fourth day after infiltration, the intensity of the interaction is very high. Here, the OLE, PDAT2, and also the DGAT2, they show the intensity of the interaction is very minimal, but on the second day, we confirm that an interaction has taken place the second day after infiltration. And the third day, very minimal, the expression has deteriorated. And on the fourth day, no interaction has occurred. So here is a diagram of what a positive reaction will look like using our control. So here, this is our YFP, yellow fluorescent protein, linked to our proteins of interest, our genes of interest, and the DS red expression vector. And this is what our control looks like when we merge the two together after day one. Day two, the intensity of the interaction is better than day one, and also on day three, the intensity is pretty good. So the results for OLE1 and PDAT2, the first day, if you can remember from the chart, the first day, the intensity of the interaction had one asterisk. So we identified that maybe an interaction has taken place. So second day, we confirm here, I know the picture is very dark, but here you can see the nucleus in the YFP and also the nucleus here in the DS red. And when you merge the two together, we can see a interaction after day two, and also after day three, we see a yellow fluorescent interaction has taken place. And also for DGAT2 here, after the first day, we see GFP, but we don't see any interaction in the DS red, so maybe the cells haven't been transformed just yet. But on the second day, the picture is very dark, I apologize. But on the second day, we can see an expression in DS red and also in YFP, which when you merge the two together, we can see a yellow fluorescent protein, yellow fluorescent image, fluorescent signal. And also on the third day, we can also see a yellow fluorescent signal in this protein here. So in conclusion, our results reveal that OLE1 interacts with either PDAT2 or DGAT2 and also showed expression only in the early stage after infiltration. This data suggests that the expression of the reconstituted YFP may be reduced to destabilization and maybe degradation of these proteins over time. So we believe that this work serves as a model for upcoming studies to determine why a protein interaction is unstable after the second day after infiltration. I would like to thank the Jaworski Lab and Dr. Jaworski for providing everything I need to do research in his lab. Shinjin, my mentor, wonderful mentor, he really gave me a lot of knowledge on how to understand protein-protein interactions. Dr. Howard Berg for the use of the confocal microscope. The greenhouse staff for providing the tobacco leaves that we use to use infiltration. The NSF National Science Foundation for funding. Also Kathy Cromer, who is absent today, Miss Sona, and Miss Leslie Hicks, and Miss Christine, RE coordinators for coordinating the program, I would like to thank you all directly. Thanks. Are there any questions? I'd like to take any questions, please. Oh, sorry, yes.
Where are these proteins localized for every ER?
They are localized in the ER, endoplasmic reticulum, and we can, using these proteins, we can find, we can have it localized any place in the cell, but by using the confocal microscope, we localize it in the ER.
Can you, when you say it is present in ER,
Mm-hmm.
is it in ER?
Yes, yes. Good question. Good question.
Thank you.
Thanks. Oh, yes, sir.
So for your biopsy assay, have you tested sort of therapy control with the single transport, one half of the YFP and the other half of the YFP that's been transported? Is that what we
No, sir. I think that is going to be further research. We basically fuse one half, like you said, one half of the YFP to another half of the YFP. But in further research, may be done to try to fuse half of the YFP and another other.
So you have a transport cell with the two constructs at the same
Oh,
time?
yes, sir.
For transport?
Yes.
So have you tested with the single transport?
Oh, no, sir.
We did not. So it's not
Yes, sir.
when you
Oh,
show the
yes, sir.
red, it has tension, yes, but not like.
Yes.
And how do you determine where you put half of the YFP?
How do we determine where we put half of the YFP? We use the N-terminal and the C-terminal of the YFP fragment, and those two combine together to create a yellow fluorescent
And you
protein.
put it on where on the protein matrix?
We put it in the, I'm sorry.
Where is it on your protein of interest?
Not sure.
Is it on the N-terminal?
Oh, sorry. Yes. Yes, we connect the half of the protein to the N-terminal and the other half to the C-terminal.
Of your other protein?
Yes.
Is there like crystal structures that dictate where you're putting your tag because you think it's going to interact that way? Or membrane bound?
No.
Are you trying all the options?
We in this experiment we just tried to link half of the half of the protein of the N-terminal and linked it to the other half of the C-terminal to each protein. So we use the OLE1, and that was linked to the N-terminal, and the PDAT2 was linked to the C-terminal. And when we had interaction, those both showed the interaction when they combined together. Does that answer the question? No? Okay. Yes.
Do you think biopsy is a better method for testing interactions, and if so, why?
Yes. I believe that in order to determine if an interaction has taken place, then biopsy would be a great method. But biopsy doesn't test the quantity of the expression being taken place. So that might, another method for testing interactions may be needed for that. But biopsy just validates if a protein interaction has taken place or not.
So let me rephrase the question. So children already detected these interactions by using nitrate. Why do you need to validate it?
We need to validate it because some interactions that may be tested in biopsy might not show in yeast 2 hybrid. So we use the biopsy analysis just to validate yeast 2 hybrid. Now there may be some, we found interaction in yeast 2 hybrid that couldn't be validated in biopsy. So it just kind of depends on what method you choose to use to validate protein interactions. Yes.
So what other method do you think?
The yeast, we could choose the FRET, the fluorescence resonance energy transfer, and we can also choose the bioluminescence resonance energy transfer, and the Western blot analysis, and the cold immune precipitation method. Yes. Good question, awesome. Thank you.